Hello and welcome to this week's What Were They Thinking? Among the many news items we have for you this week is the relationship between COVID-19 and a HIV vaccine, the future of viruses as they relate to cancer, a weird relationship between some smokers not developing lung cancers despite a lifetime of behavior, gene editing of tomatoes to provide for greater vitamin D, acidification of the ocean and oxygen production, and an AI that can begin to understand what animals are doing in the wild when they communicate. The timestamps for these and other news items can be found in the description box given below. Let's start with how COVID-19 relates to the HIV vaccines that have been in development over the last 10 to 20 years. It might in fact be better and more accurate to say how those vaccines relate to COVID-19, not the other way around. Vaccines for HIV have been in development for a very long time. They are very hard to make, and one of the most promising pathways was RNA technology and RNA-based vaccines. This is the basis upon which many of the current and most effective COVID-19 vaccines have been built. All of the research that was put into the development of RNA-based HIV vaccines before now has been given a huge boost from the COVID-19 vaccine developments. The millions, if not billions of dollars that have been pushed into developing those vaccines nationally and internationally have given HIV vaccine researchers a huge boost to where they were only a few years back. Consider how much work went into developing the structures, mechanisms, and methods for creating these vaccines. Much of that can be read as analogous to how you would develop a HIV-based vaccine. The challenge with HIV versus COVID-19 is that COVID-19 currently has a relatively small number of variants. HIV has many more. There are arguably thousands of variants of the HIV virus within the human body when you are infected. This is because over time the different cell lines begin to deviate from each other and give rise to new lineages. If you are going to develop a vaccine, it needs to be able to protect against all of these. And that means you need to teach the immune system how to recognize the basics of the HIV variant, not how to deal with just one particular variant. This has a huge upside to it though, and it does feed back into the COVID-19 situation. Currently, we have a number of variants. These variants have evolved along different lines or made different adjustments at different times. It's why, for instance, the current Omicron variant is the most widespread. If you were to try and use the original vaccines against it, they would not be as effective, but they would show some effectiveness. What if instead of vaccinating against one variant, you could train the immune system to deal with all of them simply by using one vaccine? And this is how HIV research into a vaccine for that condition can feed back into COVID-19 and create a complementary cycle of events, where by dealing with one disease, we can effectively help the development of technology for another. Infectious diseases are one thing, and honestly, they are perhaps the second biggest problem we might face that we can develop a treatment for in that manner. Another, slightly related, but not directly related, is cancer, and the issue around using various pathogens, notably viruses, to try and deal with them. Some of the most common ways cancer can occur is by certain genes becoming modified, generally being turned off or turned on. If it's a promoter gene for cell proliferation, that can give rise to cancer because it grows too often and too quickly. If it is a oncogene that's supposed to stop the proliferation of cells, they continue to grow even when they're getting signals to stop. This leads to too many cells, which is effectively a tumor. In both cases, once it becomes malignant, it is cancer. Often, some of these mutations can be caused by viruses interrupting and interfering with DNA, causing those mutations. In other cases, it is simply bad luck with the mutation occurring as they will. This is not caught by the immune system and so it isn't dealt with. In other cases, external forces like UV light can cause misconnections in the DNA strand and give rise to those same particular mutations that can cause cancer. This is where viruses can be used to deal with the very issue that they may have given rise to, by targeting cancer cells and using the same mechanisms that viruses normally use to proliferate to destroy cancer cells. That is, they insert themselves into the genome, get replicated so much that the cell literally lyses under the forces and pressure of that viral infection. The difficulty is targeting the infection and making sure that once it does its job, it goes away and doesn't start infecting other off-target cells. To do this, researchers have taken a modified pox virus and made it so that it targets the cancer cells in particular. This has led to something called vaccinia, or CF33-HNIS. It's a specific virus that is called an oncolytic virus that is designed to only infect cancer cells and avoid those that are healthy. In order to establish the effectiveness and the safety of it, they've taken 100 participants who have already had 2 types of treatment for a solid or metastatic tumor and have been unsuccessful. They will now be given low doses of this treatment via injection or IV. They will then be monitored to see how well they tolerate the injections, whether or not there's any particular side effects, if there's any existing co-therapies that might be effective, and if they could use antibody therapies on top of this to try and get an even better result. One part of this that makes the whole process much easier is that the virus that's being tested includes human sodium iodide symporter. This is a protein that helps to make imaging that much easier. Since we're talking about metastatic and solid tumors, you can measure things like density, the reaction of the image, and from this figure out whether or not you're having a positive, negative, or neutral effect. The same virus may also give rise to mutants who will have superpowers, but we can only hope. While on the topic of genetics and genes, we can now go back in time and look at an ancient sample from about 2,000 years ago. It's a human remains sample from Pompeii. Pompeii being the Roman city that was destroyed by a volcanic eruption. That eruption had the downside of destroying Pompeii. The upside is that it has created what is effectively a time capsule of a Roman city in a very, very narrow time frame. We can see the structures, the lives, the food, all of these things preserved. One thing that we can look at that is especially useful and interesting, and that's where this study comes in, are the genetics of the individuals living in the city. Most of the people who were there died from either the intense heat or the surges that caused deoxygenation. This effectively suffocated everybody in the city as ash rained down and they were buried and preserved in it. The individual that was examined in this particular study found a number of interesting things, but one was that they were infected by tuberculosis. This is one of the diseases that you may not have really heard of before now. It is something we have almost entirely eradicated from the developed world and are working to remove from the developing world. The DNA involved also indicated that the man was likely to be an Italian and from the mainland, but did have some lineage going back to Sardinia. They did also try to extract DNA from a second sample, but were unsuccessful. Still on the topic of genetics, but moving across to relationship between smoking and lung cancer. The reality is that more than 90% of lung cancer deaths in America, and this is similar for most countries, involve the use of tobacco products, generally smoking tobacco. This means that it has a very close relationship with people who die from lung cancer, but the vast majority of smokers don't develop lung cancer. This creates a strange scenario where you have a very strong relationship, but there is also a very large part of the population it can't explain. That kind of modeling is really important to figure out what's going on. We can tell you smoking is dangerous. We can show you that if you get lung cancer, it's related to smoking. What we can't say is that smoking always causes lung cancer. It can, of course, cause a variety of other cancers, but focusing specifically on why lung cancer seems to be an odd scenario, it appears to be down to the genetics involved. This, of course, doesn't mean that if you have a gene screening done and find that you have these genes, you are protected. It just means you're less likely to develop lung cancer. And it seems the genes responsible are those involved in repairing genes and preventing them from turning into cancer genes. This generally indicates that if you have certain behaviors, like smoking, you have less chance because your genetics protect you. But protection is not universal. What does appear to be common across all the people that were studied is that after about 23 years of smoking, the risk reaches a plateau and no longer continues to increase. The theory is that those who haven't developed cancer by this point have found ways to control it and mitigate any damage arising from smoking. The reality is that lung cancer and smoking are one of the more closely related factors, but other factors that do play a major role are things like diet, environment, physical activity levels, your stress levels, and more. While we talk about things like this, diet is perhaps one of the bigger ones, especially in most countries where people who smoke tend to have poorer diets. This is not just poorer in terms of financially worse off, but generally less nutritious and focus more heavily on sugars. These sugars provide the calorie content and flavour, but not the necessary nutrients, vitamins, and minerals. Most of this is overcome through labelling, but literacy on that labelling isn't always great. You may look at your standard nutrition label and think that you are drawing a comparison between two equally good or bad products. That's because most of these are standardized. Not all products are so equivocally standardized. For example, cannabis. Cannabis generally doesn't have much in the way of clear packaging requirements. It doesn't have to necessarily tell you everything that there is that you should need to know about what is in your cannabis product, whether those are brownies, biscuits, smoking cannabis, anything. The exact requirements for the labeling aren't established. That is why a study that looked at a little under 90,000 cannabis-based products from 6 different states in the United States of America has identified that there might be some rather big concerns in that area. One of the things to consider is the presence of terpenes, or the primary aromatic compound, and one of the pharmacologically active compounds when you talk about cannabis. There are also some rather serious concerns around the question of quantifying the amount of THC and CBD. These are the better known recreational drugs within cannabis that have the desired effect, let's say. The article goes into some more detail on the 3 different categories they seem to find. One of these was that they were consistently inconsistent, which meant you could never get a reliable result. Another group, and it was the largest, was a surprisingly consistent, but not necessarily particularly different, range of samples that all seem to claim to come from different cultivars. This means that although you may be expecting to get something different, the end result is fundamentally the same at a quantification level. The researchers attribute this very much to the reliability of the cultivars and a sort of self-regulation within the industry by growers. Going from one kind of crop to another, and the possibility of creating genetically modified tomatoes to increase the amount of vitamin D available. Vitamin D is the necessary vitamin that gets activated by sunlight, and from this it's able to provide a number of important bodily functions. These can be involved in heart health, neurological, blood vessels, heart, most organs you name in the body. Your average adult requires 10 micrograms a day. This is a relatively small amount, but isn't always able to be sourced and consumed. More often than not, because vitamin D has to be consumed amongst many other products, and it can be somewhat difficult to eat enough of those to get your recommended daily intake. The leaves of the plant that was being modified here, the tomatoes, had 600 grams of the precursor of vitamin D by dry weight. The researchers point out that by doing this, they are able to achieve several great outcomes. One of which is that they can, by providing tomatoes with extra vitamin D, provide enough of that and at a very low cost to help with up to 1 billion people across the globe. The other advantage is that the tomato leaves can be taken and processed to create supplements. This is a part of the plant that would under normal circumstances be considered a kind of waste. It has no real value to the farmer or anyone else. It can be theoretically recycled through composting, but that's an extra layer and it's more of a hassle. This adds value to the plant beyond just the fruit. Going from genetically modified food and the environment in that respect to the environment and smallest organisms that are responsible for production of oxygen. You may think of trees when you think of oxygen production, but they are not responsible for the vast majority of oxygen on the Earth. In fact, arguably, you have things like phytoplankton, you have things like algae, and then you have trees. They are towards the lower end of your major contributors. The tiny organisms that are called diatoms come under the heading of plankton. What they do is they take carbon dioxide and use it to build up their body, much like a plant would. In doing so, they produce oxygen and as much as 20% of the global oxygen supply. Climate change provides a hazard to this production, specifically through the acidification of our oceans. Diatoms rely on a protective shell. These protective shells tend to be made up of minerals that can be dissolved by acid. As you can imagine then, if you acidify the ocean, these creatures don't live anywhere near as well, if at all, thereby reducing the amount of oxygen that they can produce. In theory, we could see up to a 20% reduction in their volume by the end of the century. If we have 20% of our oxygen made by them, and we lose a quarter of that, it is 5% of oxygen production, as it is now. The way this happens is somewhat similar to what happens in the human body. As CO2 is generated, it is dissolved in water, primarily seawater. From here we begin to see more hydrogen ions being generated through its reaction and acidification of water. This then leads to a decrease in the carbonates that are within the water. These carbonates play a big role in that shell that these creatures form. The less calcium carbonate, the less available means there is for them to create more and new diatoms. To some effect, you can see this exact same process when you mix bicarbonate and vinegar. Bicarbonate is fundamentally the same sort of thing. All you're doing is using a somewhat more purified form. Mixing it with vinegar and acid, you create a reaction that reverses what we're talking about because it creates CO2, rather than drawing in and getting rid of CO2 from the atmosphere. While still on ocean-based topics, but also linking this back to last week, it's the development of an AI that can detect the different calls of animals in the ocean. As you can imagine, within the ocean there is a lot of noise. Some of this is background. For example, movement of waves. Others are animals actively trying to communicate with each other, the calls of whales, dolphins, and so on. There is also movement of their body, the movement of water around various structures which creates turbulence, vessels within the ocean. All of this can interfere with the ability to isolate and understand any given call from a single animal, let alone beginning to detect the calls of animals swimming in the ocean, separate them out, and begin to figure out what's happening. This particular AI currently focuses on dolphins, and it relies heavily on a machine learning algorithm that's intended to categorize different sounds and then put these in context of what it knows they mean. This is a very difficult thing to do, for all of the reasons mentioned, let alone actually making it work. Where this adds yet another layer of benefit is that it is a particular approach that was developed not to deal with marine animals. Rather, it was developed to deal with the ultrasonic sounds that mice and rats make. This means that it already has use outside of the ocean. More importantly to those two big points is the nature of the program. It's called Deep Squeak. It is described as a user-friendly, open-source tool for analyzing a variety of terrestrial species. This means, at least in theory, it could be used by both researchers on a tight budget, those who are trying to encourage the use of open-source tools, and for those who are trying to work with communities for what are called citizen scientist projects, which is really just community outreach with a more fancy name. Next we go to a crab, but not a crab that is real, rather a technological crab. It is a tiny, remote-controlled robot that can walk. The robot is arguably smaller than a pencil lead, at about 0.5 millimeter or less than 1/50th of an inch. This is why we don't use imperial numbers when you have to go to fractions of a particular thing. The robot itself, as it is designed, has many uses. The big ones are going to be things like observation of machinery, getting into confined spaces for monitoring and repairs, surgery, and more. The smallest robots have the most use. This is why nanobots have been depicted doing so very many things within science fiction. The robot itself moves by a change in its substrate, and this allows it to move about 1/4 millimeter every second, or its own body length or about 1 millimeter every 4 seconds or thereabouts. The structure is kind of like an origami piece. It folds and moves based on the movement of what occurs underneath it. As different parts are pulled in the substrate, they allow the material above to relax or tension. This means that those parts will either move forwards, backwards, or, as required, elsewhere. When you can set up the layers to move in a particular way, you can then make it move, and you can move it in a very controlled manner. It's almost like the memory forming pasta we've mentioned in past videos. There's also been other instances where they've used the different folding techniques to create a single layer structure that came out of what is basically a flat pack and self-assembled. Where they relied on the ability to be unpacked and self-assemble, this does need some input, and that's an important caveat to this. The way you give that input is through lasers. Those lasers are what affect that substrate and either relax or restrict it, and what either make it move forwards, backwards, or elsewhere as required. That does, however, create one small issue. They do get hot, but because they're so small, they also cool down relatively quickly. This means that you can impart messages frequently and quickly. By doing so, you can get very fast reactions, and you don't need to worry about the heat issue as much. But it is one of the considerations, and will be slowly counteracted as miniaturization goes further along this pathway. The smaller the object, the more easily it exhibits its heat and puts it out into the local environment. Our last piece of news is probably something that should be taken as a prank, but it's the observation by NASA of an undersea volcanic eruption. NASA has rather humorously named this Sharkcano. This is not a new suggestion to replace Sharknado. The volcano was found near the Solomon Islands in the Pacific Ocean. The area is already somewhat active as far as volcanoes go. It has been given the name Sharkcano largely because of the surprising population of hammerhead and silky sharks in the crater that is there. That's all the news we have for you this week. Thank you for watching. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.